Okay, this screencast is basically going to be a review of DNA, RNA transcription and translation, mainly because in order to understand how COVID infects our cells and replicates itself, you need to have some knowledge of especially transcription and translation. And we've already been through this back when we covered the information having to do with the sickle cell anemia case, but I want to review it for you just to make sure that you're remembering everything that you need to know to understand what it is that COVID is doing when you come into contact with COVID. So once again, we're going to just review how transcription and translation are involved in viral infection pathways and explain how the reproduction of the virus differs from transcription and translation in eukaryotic cells. And it's basically going to explain how viruses utilize the normal functioning of your cells to make copies of itself. So just to remind you, nucleic acids are made up of nucleotides. Nucleotides have a phosphate, a sugar, and a nitrogenous base. If it's DNA, you have an H, a hydrogen at the number 2 carbon. If it's RNA, you have an hydroxyl group at the number 2 carbon. Deoxyribonucleic acid is deoxy because it's lacking this oxygen. Remember that there are different kinds of nitrogenous bases, single ring bases and double ringed bases. Double ring bases are purines, single ring bases are pyrimidines. I always remember it because pyrimidines have the long name, but they're the smaller molecule. Purines have the small name, but are the larger molecule. And remember also that guanine always bonds with cytosine, and thymine and uracil always bond with adenine. This is adenine over here in the corner. So these molecules of molecules of ribose have carbons that are numbered from carbon 1 to carbon 5. So carbon 1, carbon 2, carbon 3, carbon 4, and carbon 5. And you always add new nucleotides to the 3' carbon. So this phosphate is going to be added to this carbon right here. And so there's this directionality to DNA. And so on one strand, it's going from the 3' to 5' end, and on the other strand, it's going the opposite direction. And in the case of DNA and RNA, DNA is always a 2-stranded molecule and RNA is a single-stranded molecule. And so when we think about making copies of DNA, we have to make copies of both strands of the molecule, whereas RNA is built from a structure of DNA, using DNA as a template, but it is only building 1/2 of the strand, and it also has a different sugar. So DNA encodes information for the structure of amino acid sequence, which are going to ultimately be proteins. The sequence of nitrogenous bases dictate what the sequence of amino acids will be. It's a double helix, which means that you have these 2 strands and they're always combined with one another and bound to one another using these base pairing rules, such that anytime you have an adenine on one strand, you're going to have a thymine on the other. Anytime you have a cytosine on one strand, you're going to have a guanine on the other. And once again, there is this directionality to the molecule such that one strand moves from 5' to 3', while the other strand is oriented in the opposite direction. Adenine and thymine bonded, cytosine, guanine bonded, adenine, thymine bonded. Notice that there are different numbers of hydrogen bonds between cytosine and guanine versus adenine and thymine. This is another thing that helps them not mess up when it comes to do complementary base pairing. So when we think about how we go from DNA to protein, we can think about the central dogma, where taking a DNA molecule and using that DNA molecule to specify a messenger RNA molecule, that process is transcription. Then we have taking that messenger RNA molecule, using a ribosome to build an amino acid sequence. That process is called translation. But then we also sometimes need to make copies of our DNA because we need to go through cell division. This process over here is called DNA replication, where we take a single strand of DNA and we create 2 new strands of DNA from that single strand of DNA. So think about replication first. We have to open up the strand of DNA, and then we use these enzymes called DNA polymerases to basically add new nucleotides to an already existing half of a DNA molecule. And from this, we get 2 new DNA molecules. Each of these DNA molecules is made up of half old DNA molecule and half of it is new DNA molecule. And so in this way, this is called what we call semi-conservative replication, because we're always using an original DNA molecule as a template for building the new molecule. Once again, this should hopefully be a review for you. When we build these things, you have to build them in a 3' to 5' sort of way. So if we were to say, well, this is the 3' end of this molecule over here, that means that this is the 5' end if you follow it through all of this, which means that this must be the 5' end of this original molecule, which makes this the 3' end of that molecule over here. And so when we're adding new DNA, it's easy on this strand because this is the leading strand, because you're always just adding new nucleotides to the 3' end because it's the 3' end that is always exposed. But in this case, if you start adding molecules to this, you're trying to add molecules to the 5' end. And so what the cell has to do is it takes this lagging strand and loops it around. And then as a result of looping it around, if we look at the original template strand, 5' to 3' is oriented this way, but as we loop it around, that then allows us to add new nucleotides, where we add new nucleotides to the 3' end of this developing new thing. But what we have to do is we have to do that in small segments, which leads us to these little fragments called Okazaki fragments. So we made this new DNA, but we made it in this direction, going back towards the 3' end. And then we're making a new strand of DNA here. And when you get to this point, you can't make it anymore. And so this thing is going to go, this DNA polymerase is going to go and latch on to the lagging strand further down the line. And then it's going to create another Okazaki fragment. And then after those Okazaki fragments are all formed, then there's an enzyme called ligase that comes in and stitches those fragments together. So we are constantly making new copies of DNA from old DNA at sites of replication, and there are multiple sites of replication, and they move in both directions, and that enables us to replicate our DNA really quickly. So that was DNA replication. That's taking DNA polymerase and using DNA polymerase, which is an enzyme, to take a single molecule of DNA and create 2 new molecules of DNA. Transcription is involved in taking a double-stranded molecule of DNA and creating a messenger RNA strand. And once again, this is done according to the rules of complementary base pairing. RNA strands are built on a 3' to 5' adding the 5' end of one nucleotide to the 3' end of the developing molecule. And after this is all done, DNA reforms. And so there are 3 phases of this: the initiation phase, the elongation phase, and the termination phase. DNA replication differs from RNA transcription in that we start with a double-stranded molecule of DNA, we split that in half, we add new DNA nucleotides to the 2 halves, and we end up with 2 new strands. In RNA transcription, we start with a double-stranded molecule. We open it up, revealing the template strand, and then to that template strand, we add RNA nucleotides to create essentially a messenger RNA molecule. And then the messenger RNA molecule goes through post-transcriptional processing to give it a 5' guanosine cap and a 3' polyadenine tail. So in eukaryotic transcription, we basically have initiation where an RNA polymerase will bind to a section of the DNA, which represents the beginning of a gene. It will open up the DNA and it will start creating messenger RNA. When it gets to the termination site, it will stop creating that messenger RNA, and that messenger RNA is then ready to be processed. In order for this to work, though, you have to have this thing called a pre-initiation complex forming. It's a series of transcription factors or chemicals or compounds that have to bind to the DNA just upstream, just before you get to the promoter site. And only after these transcription factors have bound here will they then recruit the RNA polymerase. Once that has happened, then you have the entire pre-initiation complex present. But just building that infrastructure doesn't prompt the replication of, or the transcription of a messenger RNA molecule. You actually have to have another section of the DNA, sometimes hundreds or maybe even thousands of DNA base pairs upstream of that, this area called an enhancer that is going to attract compounds, chemicals to it that will then interact with the transcription factors. And when those activators and mediators make contact with these transcription factors, that kicks off the process of transcription. So you can assemble this pre-initiation complex, but if this enhancer is silenced, you won't ever get transcription happening. We saw this in the locus control region of the sickle cell site. Basically, there's a region upstream of fetal hemoglobin. That region essentially turns on fetal hemoglobin. Normally, when you're an adult, that locus control region is blocked from fetal hemoglobin by a particular protein that binds to the promoter of fetal hemoglobin. So this locus control region will go over here to adult hemoglobin and turn adult hemoglobin on rather than turning fetal hemoglobin on. And they cured sickle cell, at least got rid of the symptoms of sickle cell, by basically getting rid of that protein. So RNA polymerase is making the messenger RNA molecule by opening up the DNA double helix and then adding RNA nucleotides to this developing RNA transcript. So when we think about the structure of a gene has this promoter region where the gene is turned on, a series of DNA nucleotides, a long series of DNA nucleotides that are coding if they're exons or non-coding if they're introns, and then you get to this termination sequence. But way upstream is this distal control element that essentially turns on the switch here. After the messenger RNA is transcribed, we have a pre-messenger RNA transcript, and then that transcript gets spliced by removing the introns so that you're just left with coding sequences. And then the other thing that happens to messenger RNA after it's formed is you have this 5' guanosine cap and this 3' polyadenine tail. This structure I want you to pay careful attention to because it will become important when we start thinking about COVID-19, which is a coronavirus, which is a type of RNA-based virus. It has RNA as its nuclear material. So now we take this messenger RNA molecule that we formed, we ship it out to the cytoplasm, and we make a protein. And so there's a specific set of rules for this. The DNA is complementary base paired to form RNA, and then this RNA is arranged in 3 nucleotide sequence called codons that then dictate what type of amino acid is going to be placed in that sequence of amino acids making up a protein. Once again, thinking about amino acids, they're made up of an amine group and a carboxyl group, and then this variable group. This, once again, provides directionality to a protein molecule, and you're always adding new amino acids to the carboxyl end of a protein. We take 2 amino acids, we take this water molecule out of the middle, that's a dehydration sequence, and then we form this peptide bond. So we've added the nitrogen, the amine portion of one amino acid to the carboxyl portion of another amino acid. So we're adding amino acids to this end as we build this. And so there is directionality in the same way that there is directionality in a DNA and RNA molecule. The codon table specifies what the sequence of codons are in messenger RNA and what amino acids they correspond to. So if you know the structure of the DNA, that dictates the sequence of nitrogenous bases in RNA, and that sequence of nitrogenous bases in RNA essentially dictates the sequence of amino acids in proteins. Methionine is what starts. AUG, the start codon, is what starts the process of protein synthesis, and there are 3 stop codons that terminate it. So we talked about protein structure, come in the single chains of amino acids. These chains can take on 2-dimensional structures. These 2-dimensional structures can then become wrapped around one another. And then when you have multiple proteins all wrapped around themselves, then that is quaternary structure. This is the structure that we see in hemoglobin, where you actually have 2 alpha hemoglobin and 2 beta hemoglobin strands. So there are 4 proteins that make up the overall molecule of hemoglobin. That's a quaternary structure. Amino acids have different characteristics, especially where charge is concerned. So some things are uncharged, other things have negative charges, other things have positive charges. Translation, you need an RNA transcript, you need transfer RNA, and you need a ribosome. Once again, it has 3 steps: initiation, elongation, and termination. The mature RNA transcript has this structure, a 5-methylguanosine cap, which is important to signaling the ribosome where the beginning of this molecule is, and then the poly A tail, which is hundreds, in many cases, hundreds of adenines long, and this basically stabilizes the molecule so that as the molecule begins to break apart, it's not breaking apart the coding sections, it's just breaking apart this long polyadenine tail. Transfer RNA takes the amino acids that are up here at this portion of the molecule. It uses the codon structure of the messenger RNA molecule to allow the anticodon to complementarily base pair to the messenger RNA. And the way this proceeds is you have 3 sites in your ribosome. So the ribosome comes together at that end where the 5-methylguanosine cap is, and there's a large subunit and a small subunit. The large subunit has these 3 docking sites, E and P and A, and the first transfer RNA molecule is going to come into this P site. It's going to complementary base pair with the messenger RNA, and it's going to have an amino acid attached to the top of it. So this is the large subunit. This is that first transfer RNA. It is complementary to AUG, adenine uracil guanine, so it's uracil adenine cytosine, and it will have a methionine up here at the top. That forms this, that causes the ribosome to form up on the messenger RNA, and then enables a second transfer RNA molecule to come in here. It will complementary base pair with the next codon, and that methionine will be bonded to whatever amino acid is there. Once that has happened, this whole thing shifts down 1 codon such that the original transfer RNA molecule moves to this site, the E site, which gets ejected. This transfer RNA molecule that was in the A site is now in the P site, and a new transfer RNA molecule comes in to the A site so that you can bond the pink amino acid to the green amino acid. And this continues on and on and on until you've built an amino acid sequence, and then the stop codons correspond to transfer RNA molecules that lack an amino acid. So there's nothing to bind this green molecule 2, and so that terminates the process of protein synthesis. So, what does this have to do with being infected by COVID? Well, what it has to do with is SARS-CoV-2, which is right here, has a bunch of proteins on the outside of it. These proteins are called spike proteins. And these spike proteins bind to these kinds of proteins in the plasma membrane of your epithelial cells in your lung. And that receptor is called ACE2. It stands for angiotensin converting enzyme 2. ACE2 normally just interacts with angiotensin, which is a type of hormone that regulates blood pressure. But in this case, the spike protein of the COVID-19 virus binds to that receptor, and that receptor engages in receptor-mediated endocytosis. And so we saw last screencast that what this does is it essentially tells the cell to create a vesicle and bring that virus into the inside of the cell. Once it is inside the cell, it breaks down the virus and releases the RNA of the cell. And the RNA of the virus. And the RNA of the virus looks very much like messenger RNA. So this is viral RNA. It has a 5' methylguanosine cap. It has a 3' polyadenine tail, and it has a bunch of coding sequences in between. The first two of these coding sequences code for a set of proteins, and these proteins are specific to creating copies of viral RNA. So remember back when we talked about replication of DNA. Replication of DNA involves DNA polymerases that basically take the two strands of DNA and create a two-stranded molecule out of a one-stranded molecule. They create two new DNA molecules out of a two-stranded DNA molecule. In transcription, there are RNA polymerases that allow you to take a two-stranded DNA molecule and create a one-stranded RNA molecule. So eukaryotic cells, which is what this is, have the ability to take two-stranded DNA and make new copies of two-stranded DNA, or they are able to take two-stranded DNA and create one-stranded RNA out of that. What they don't have is they don't have replicating mechanisms to take one-stranded RNA and make copies of single-stranded RNA. And so what happens is when the cell brings the virus into it and breaks it down, it releases this RNA. This RNA, which looks very much like messenger RNA, is then going to be translated into two proteins, PP1A and PP1AB. These two proteins make up this thing here called the replication transcription complex. This is a set of proteins created by the ribosomes of the eukaryotic cell. So this is eukaryotic cell ribosomes that are basically translating portions of the viral RNA to create these two proteins that go into making this replication transcription complex. That replication transcription complex actually allows the replication of new viral RNA from RNA. And so it's an RNA-dependent RNA polymerase rather than a DNA-dependent RNA polymerase. So it's a different type of polymerase than the type of polymerase that your cells contains. Once that's made, it can then take this viral RNA and make copies of it. It makes copies of that RNA independent of any of the machinery of the host eukaryotic cell. Those copies are then used to begin putting together new versions of the virus. So you make copies of the viral RNA. That viral RNA also has some other structural components, such as making the capsid, the capsule that contains the RNA, and making things like the spike proteins that are on the outside of it. And eventually, the eukaryotic cell creates these proteins, packages that virus up, and ships that virus out. And so the virus basically tricks the cell into bringing it in. The cell breaks it down because it recognizes it as something that doesn't belong, releases the RNA. The RNA is similar enough to messenger RNA that ribosomes of the host cell will make viral proteins that then make copies of viral RNA. That viral RNA is then used to build the capsule that is going to then encompass the viral RNA, and then those virus particles will be exocytosed out of the cell. And so you've basically turned your cell into a factory that creates new copies of the virus. And so that's how infection happens. In the next screencast, we will talk about how your body mounts an immune response.